Hi everyone. In this video, we will learn what is monitoring and control in a fermentation. Now, microbial fermentation is going on. You have selected a fermenter, you have prepared medium, you have sterilized the fermenter and medium both. You have prepared the inoculum because you are not going to inoculate a looful of the inoculum in such a large fermenter. So you have carried out inoculum preparation. You have inoculated your culture. Now your fermentation, if it's a batch fermentation, let's say goes on for 7 days. Now during the 7 days of the fermentation, the temperature, the pH, the degree of agitation, oxygen concentration, dissolved oxygen in the medium, and other factors have to be kept constant during the fermentation process. If I talk about temperature, there's an optimum growth range of each organism. You should be knowing at what temperature your process organism grows best and produces the product in large concentrations. We have already determined that in flask, in laboratory fermenters, in pilot scale fermenters during scale up, you also know what is the pH which is best for growth and best for the production. The degree of agitation, dissolved oxygen, all these factors have been determined for a fermentation, and these factors have to be kept constant during the fermentation process. Now, I inoculate the culture in a fermenter. Let's say if it goes for 7 days. After inoculating the culture, I may go home and sleep that the organism will do its job and will produce the product during the 7 days. It's not like that. There has to be a monitoring system that monitors the temperature, pH, degree of agitation, oxygen concentration, and all the other factors during those 7 days. Now, the provision of such conditions requires careful monitoring so that any deviation from the specified optimum might be corrected by a control system. So to continue with the same example that the fermentation is of 7 days, and let's take example of one parameter here to explain what is monitoring and what is control. Let's say temperature. Now, this is the fermenter, let's say, and this is a huge industrial fermenter in which the sterile medium is already present. It has been inoculated at day 0 and the fermentation has started. Now, the organism that you have inoculated has an optimum growth temperature of 31 degrees Celsius. Now, this is the temperature which is best for the growth of your process organism and also best for the production of the product. So you have to ensure that the temperature remains 31 degrees Celsius during those 7-day fermentation. The temperature should not be decreased from 31 to let's say 25 or 24, and the temperature should not be increased from 31 to let's say 40 or 45. If it increases so much, forget about the production. Your organism may stop growing or may get killed if it is mesophilic. So much deviation is not accepted. So let's say you have a monitoring system, a thermometer or a thermistor, which is in constant contact with the broth, and it monitors the temperature of the fermentation or the temperature of the broth or medium during those 7 days. This monitoring system, which is thermometer or thermistor in this case, this will tell you that the temperature is 31 during the 7 days. Okay, and if the monitoring system tells you that the temperature is not 31, it has gone up to, let's say, 34 or 35, there has to be a control system which should bring back the temperature from 34 to 31, which is your original temperature, which is best for the production and growth. So this is monitoring and control. So monitoring is monitoring whether the temperature is 31 or no during the 7-day period. And if there is deviation, which is 34 in this case, that deviation, okay, has to be corrected by a control system so that the temperature comes back to 31. This is monitoring and control. Now, the monitoring of a process may provide information on the progress of the fermentation. So monitoring is very important, because if, let's say, if I took example of temperature, temperature increases too much, if it keeps on increasing 35, 40, 45, 50, it may kill your organism, and then there's loss of fermentation. So monitoring is important, but it may also tell you on the progress of the fermentation, and such information may indicate the optimum time to harvest, when to stop, and whether the fermentation is progressing abnormally and there is some contamination or strain degeneration. That also can be obtained by monitoring the process. Now, how would you monitor the process? Using sensors. In the previous example, the thermometer or thermistor, the example that I gave, that is a sensor. So it's monitoring, it's sensing. So there are three main classes of sensor. Sensors which penetrate into the interior of the fermenter. Example, pH electrodes that determine the pH, or dissolved oxygen electrode that determine the dissolved oxygen of the broth or medium. Now, obviously, such sensors have to come in direct contact with the broth or fermentation medium. They are penetrated into the interior or inside of the fermenter. Obviously, you cannot keep a pH electrode outside the fermenter and find out the pH of the fermentation broth during a fermentation. You cannot keep a dissolved oxygen electrode 2 kilometers away from the fermenter and find out what is the dissolved oxygen in the broth. So these sensors have to be inside the fermenter. So they penetrate the interior of the fermenter. Then there are sensors which are not inside the fermenter, but they operate on samples which are continuously coming out from the fermenter. Example, exhaust gas analyzers. Exhaust gas analyzers analyze the exhaust gas which is coming from the fermenter. And then there are sensors which do not come into contact with the fermentation broth or gases. These sensors, the third type, they are neither directly part of the fermenter or integrated into the fermenter, nor they come into contact with the broth or gases. Example, tachometers, load sets. Now, these sensors can also be defined in relation to its application. Inline sensor. What is inline sensor? Inline sensor is integrated into the fermenter and it is used directly for process control. So this inline sensor is a sensor which is integrated into the fermenter. It's part of the fermenter. Okay, and it is used directly for process control. So for example, if let's say a sensor that determines the pH, or a sensor which is sensing the foam. So the sensor will sense that there is foaming or the pH is changed, and automatically the thing would be corrected or controlled. So this is such a sensor is called as inline sensor, which is integrated into the fermenter and it is used directly for process control. The next is online sensor. Now, what do you mean by an online sensor? Online sensor is also integrated into the fermenter. So you would say this one is similar to inline. No, it is integrated into the fermenter like inline sensor, but the values cannot be used directly for control. So in an inline sensor, the first one, the sensor is monitoring, and if there is any deviation, automatically it would be controlled as well. But in the online sensor, it is integrated into the fermenter, it's monitoring the parameter, but the values cannot be used for direct control. Someone has to, come and manually at least enter the data into a computerized system for control. So in online sensor, the system would tell you that there is deviation. So if I take back the example of the temperature, the online sensor is integrated into the fermenter, it's monitoring, it will tell me that the temperature is changed from 31 to 34, that's it. But the sensor would not automatically control it. Someone has to go manually and enter the values into a computerized system that the temperature is 34 now and the original, or it should be 31, so that there is control. The last sensor or the third sensor is the offline sensor. It's not integrated into the fermenter. unlike inline and online, and it cannot be used for process control. This is, most of the things are manually here. So it is not integrated into the fermenter. It's not part of the fermenter offline sensor. Someone has to go and monitor a parameter. Someone has to go and measure something, dry weight analysis or wet weight analysis, or to take a reading. And then if it is not correct, then another step has to be taken for control. So manually the monitoring is done, and then again control has to be done. So offline sensor is not integrated into the fermenter, and it cannot be used for process control. This was all about monitoring, control, and the sensors.